Hello everyone, my name is Han Chang. I'm a product manager in microbial. In this presentation, I'd like to introduce our paper published in Meta, which is entitled MIST, a microbial identification and self-tracking system for next-generation sequencing data. The Professional Committee of Microbiology of the National Pharmacopoeia Commission organized the drafting of the technical guidelines for microbial whole genome sequencing, aiming to standardize the method, process, and technical indicator of microbial WGS and ensure the accuracy of identification. Based on the guidelines, we developed an integrated microbial identification system designed to meet the needs of microbial identification and contamination investigation in food and drug quality control. MIST integrates 3 analysis pipelines, 16S, 18S, ITS applicant-based microbial identification, WGS-based microbial identification, and SNP-based microbial self-tracking. MIST can analyze sequencing data in a variety of formats, such as FASTA, BCL, FASTQ, etc. It can be connected to a high-throughput sequencing instrument to acquire sequencing data directly. We also developed a publicly-accessible web server for MIST. Whole genome sequencing, amplicon, and metagenomic sequencing are increasingly used in research to produce complicated environmental sequencing datasets, which paved the way for cultivation-independent genetic content assessment and exploitation of the entire communities of organisms. Therefore, it is urgent to develop WGS and amplicon-based microbial species identification pipelines in the field of food safety and drug control. Here, we developed a system to analyze the WGS amplicon sequencing data for microbial identification, MLST typing, and SNP self-tracking. In our system, one important potential use of the WGS microbial identification pipeline is to identify contaminated sequences or metagenome samples. It has great value in speeding up the pathogen detection in clinical laboratory, while the existing identification and taxonomy methods may be unreliable with contaminated samples. Finally, you are welcome to publish your work in Meta.